Hello and welcome to Conversations with Dr. Bachner. Once again, it is Howard Bachner, Editor-in-Chief of JAMA. And I'm joined by Adam Lauring. Adam is a physician, a PhD. He's an associate professor of medicine in the Department of Infectious Diseases, Microbiology, and Immunology at the University of Michigan. Welcome, Adam.
Thanks for having me.
So we're going to talk about coronavirus, evolutionary biology, and these new emergence of variants. I just want to remind people who are watching or who will listen, Adam published a viewpoint with us now almost a month ago entitled Genetic Variants of SARS-CoV-2, What Do They Mean? And then more recently, John Moore and Paul Offit have followed that with an additional viewpoint entitled SARS-CoV-2 Vaccines and the Growing Threat of Viral Variants. So Adam, for my listeners and maybe to benefit me, let's start with some basic questions. SARS-CoV-2 is an RNA virus. What does it mean that it's an RNA virus?
Yeah, so at its basic, it means its genome is made of RNA and not DNA. But functionally, what it means is RNA viruses tend to have higher mutation rates than DNA viruses, and that's because the enzymes, the polymerases that the virus uses to copy its genome, tend to make more mistakes when copying the genome, and those are the mutations. And interestingly, SARS-CoV-2, like other coronaviruses, they're sort of a unique family in that they make fewer mistakes than most other RNA viruses because they have what's called a proofreading mechanism. They have a protein that recognizes a mistake and corrects it. So they, at a biochemical level, they make mutations less frequently than other RNA viruses.
What would be examples of other RNA viruses and DNA viruses?
Sure. So other RNA viruses of note are influenza virus, poliovirus, measles, mumps, hepatitis A, hepatitis C, and then there's the retroviruses, which have RNA genomes, but they behave a little bit differently in how they replicate. And then DNA viruses would be herpes viruses, pox viruses, so cytomegalovirus, Epstein-Barr virus, herpes simplex virus, parvovirus, many others.
So has the emergence of these variants surprised you?
Yes and no. I think yes, in that if you asked me in February or March, would we be where we are today? I wouldn't think so. No, in that, if you think about the fundamentals of how these viruses evolve, it shouldn't be surprising that they would, new variants would arise. I think kind of the extent of it all has been a little bit surprising. And I think, that also has to do with the epidemiology and kind of what's going on globally with this virus.
So we'll get to that in a second. So, the new variant, the SARS-CoV-2 was identified a year ago, late December. Mm-hmm. The genome sequence is released, which is remarkable. And there's a genotype. And then there's a report that a variant has developed. We'll get to how that may occur. When they say there's a variant, what do people mean by that?
Yeah, it's one of those things that terminology is everything and nothing at once.
Right.
A variant means it's just different, right? And so, if you look at the genome sequence, it's different from that other SARS-CoV-2 virus. And so, at its base, you can say anything that's different is a variant.
Okay.
And so that's the problem, is when do we say something is a variant and when is it just another sequence that's different? And that, has to do with what we're paying attention to. And so right now we're talking about certain variants that have, increased in frequency and may or may not be concerning. And so it's a little bit in the eye of the beholder.
And how does your lab or other labs go about identifying that new variants would emerge, and why would they even test for new variants?
Sure. I think what my lab and many others do, we do sequencing of samples of viruses from people, and in the case of SARS-CoV-2, you get the sample and you sequence it, and then we compare it to other sequences and see how they're different. And initially, we were doing that a lot to kind of inform the epidemiology, because the sequences and how they change can tell you where the virus is transmitting and who's spreading the virus to whom. And that has been something we've been interested in for a long time, and so we started working in that area. But now, the sequences that my lab and many other labs around the world generate allow you to see how variants are rising, and either rising in parallel in different locations, which provide clues, or in the case of the tremendous amount of sequencing in the United Kingdom, people have been able to follow how these variants come to dominate, and across regions or across the entire country. That's very informative. And now we're trying to learn how variants are moving from one country to another using this sequence data.
So when you first detect a variant, does it immediately raise concerns that it could be more infectious, more transmittable, lead to more mortality, or do you have to combine that then with epidemiologic data to know that?
Sure, I think more the latter. I think it's always sequence data on its own can tell you things, but it's much richer with the epidemiologic data. And I think another point that I think is critical is you're only as good as your data. And one thing that has stood out in terms of the B117 story, which we'll probably get to, is the United Kingdom has done a tremendous amount of sequencing. So I think it's something like 10% of the viruses in the country are sequenced, and so that allows you that degree of sampling allows you to really understand things. Where in the U.S. we don't sequence as much, so you might see a virus increasing, but you're not sure if that's really reflecting its change or if it's something to do with your sampling. That maybe you're sampling a certain area and it's prevalent in that area, but so it's hard to infer what's going on unless your sampling is unbiased and broad,
and
like
then
it has been in the UK.
And then you have to marry epidemiologic data
Sure.
with the emergence, say, of the variant in the UK,
Right.
to understand if it's changed the epidemiology of the disease,
Exactly.
right? As a standalone, it's not very helpful.
Exactly. As a standalone, you can get a lot of clues. But yeah, it's definitely much richer with the epidemiologic, or even in some cases the clinical data that go along with it, to really paint a picture of what's happening.
Okay, so now let's take a step back. You're a virus in the world, and you're floating around, and then you begin to see people who are immunized, people who got some remdesivir, people who got some steroids, people who recovered and have antibodies. What's the virus doing to mutate so that a variant occurs? So make believe you're a Disney movie and you're the virus. What are you doing to protect yourself?
Right. So if we're gonna think of viruses as anthropomorphizing them, right? If you're thinking about what does a virus want to do, the virus wants to be able to get to the next host and be able to make more of itself. That's all a virus cares about. And so viruses are mutating all the time. Every time they copy themselves, they're making mutations. And you can think of it as trying out new solutions, trying to, that mutation is either going to make you a better virus or a worse virus. And then what you have is selection. So survival of the fittest, for lack of a better term. And the viruses that have a mutation that allows it to do something better are going to win out over time. And what we see is, well, as things change on the ground, viruses might be better at growing within certain hosts, or it might be better at transmitting from one host to another, or as you suggested, then there's going to be an overlay of people who have immunity, and so the virus may find itself in someone new in a more hostile environment because they've got immunity. And so it's going to make mutations, and that's going to change which mutations are going to be better or worse for the virus in that, little microcosm in that one host. And writ large, that's going to really change things over time.
So then, obviously, just the last 2 or 3 weeks, it's really quite amazing. It finally seems like much of the world is catching up with vaccines, Moderna, Pfizer, J&J, Sputnik, Novavax, the Oxford vaccine, mRNA adenovirus. And then all of a sudden we hear you have a UK variant, a South African variant. Now there's a Los Angeles variant that we'll shortly be reporting on. And it immediately raises the concern that is the vaccine effective against this new variant? So I've made you CEO of Pfizer. You're also an MD and a microbiologist. So how do you go about trying to understand if your vaccine is still effective against a new variant of the virus?
Sure. I think that's whatever, the $64,000 or maybe $64 million question. I think there's two parts to that as I think about it. One is, what does it mean for a vaccine to be effective? What are we measuring, right? Are we thinking about vaccine effectiveness in terms of protecting the most people in the population, protecting against severe disease or not severe disease, protecting against infection or transmission? All those are different ways of thinking about how a vaccine works and whether it works or not. And then you've got what are called correlates of protection. And that is probably the early warning sign that a lot of vaccine makers will probably look at to see, okay, what's coming down? Do we need to worry about this? The correlate of protection that people use the most are looking at serum, looking at antibodies, and seeing in vitro in the lab, mixing it with virus and see if it blocks the virus from infecting cells. It's easy. Even I can do it. And it's pretty easy to get serum from people. And so what we see is a lot of studies where you take serum, you test it with virus 1, virus 2, and see if there's a difference. And that provides you a clue of whether the antibodies that are stimulated by the vaccine protect you or not. But then there's also the factor of, well, that's just one part of immunity. And, you may not have a lot of antibodies in your blood, but you may have cells that produce antibodies, and so you may still be protected. You may have T cells. There's a lot going on. And so what you're looking for are clues with these seroassays. And, sometimes they're reliable and sometimes they're not. But what we don't know is kind of what's that threshold. So if you see in your in vitro assay that you can only neutralize the virus half as well or 1/10 as well, if it's a variant, how does that translate to whether someone is protected? And that's not clear right now. And I think we need to learn more about that as well.
If you have serum from me, say I had disease,
Yep.
or I was vaccinated, so that serum is going to have IgG and IgM in it, but it won't not have my T memory cells, correct?
Right.
And so that's what you were talking about. In some regards, it's a partial test. Is that accurate?
Yeah, I mean, it not only won't, unless, in serum, you won't have your T cells, you won't even have your memory B cells or your plasma cells that might, be important just for the antibody response. So there's a lot we're missing. It's important work, but it's important to recognize that it's a correlate. It's correlated with protection. And we still don't know in many cases how it translates to where the rubber meets the road, and is that person or is that population protected?
There's quite a few questions, but I have one or two more, and then we'll get to the questions, Adam. So we thought of I've called it the great pandemic of 2020. Obviously, people call it many other things, SARS-CoV-2 and COVID-19. Is this just going to become a chronic coronavirus pandemic at some low level? That is the fear that people are beginning to develop, that We will appropriately vaccinate against the current variants. I've said yesterday, I had a chance to talk with Tony. The variants aside, I'm guardedly optimistic for March and April. We're vaccinating 1 million. We're supplying 1.3 million vaccines plus a day. There's 55 million people older than the age of 65. Theoretically, at the current rate, everyone in that age group could be vaccinated by April 1 if they want the vaccine, or at least get the first dose. That will dramatically reduce the rate of death, which is still 80% in that age, or 80% of the deaths are in that age group. But that obviously is taking this variant and whether or not the Moderna Pfizer vaccines, ultimately the J&J vaccine, are effective against it. Are we? Is that what you anticipate? Chronic SARS-CoV-2 coronavirus disease? I
don't anticipate. I think the Super Bowl's this weekend. It's where's the goalposts, right? What do we?
Okay.
Are we eradicating or eliminating SARS-CoV-2? I don't think so. Can we get to a place where we're not, confined to homes and not able to go out to eat and, not have our kids at school? I don't think that's going to last, a super long time, months. And I think we may get to a place where, we have annual, just like we have flu epidemics every year. We might have, wind up in a place where, every year there's, a time of year when we have a lot of SARS-CoV-2 around, and some people get sick, some people get very sick, some people die, but it won't be where we're at now. That's, I think, where we're headed.
Now, virtually all of the vaccines, adenovirus vector vaccines, mRNA, have focused on the spike protein. For the few vaccines that are more traditional, heat-killed or manipulation of the virus, that would be less true. Do we need to think about vaccine development that's broader in its target than the spike protein?
Yeah, I think that's a really good question. I mean, I have worked in the influenza field for a while, and everyone there has been focused on HA, or hemagglutinin, which is kind of the corresponding protein to spike on the surface, because it's thought that protein is really important in terms of providing immunity, or targeting that for immunity. But in recent years, people have learned that this other protein on the surface called neuraminidase, or NA, is also important. And so vaccines that direct a response to that can also provide some protection. And so I think it is something that I'm sure many are looking at now in terms of other ways of combining antigens in vaccines, or different spikes in vaccines, or different ways of updating vaccines. That if, as you said earlier, if I were the head of Pfizer, I would be thinking about those, and I'm sure they are.
One last question for me there, then I'll go through the questions. Yeah. The holy grail in influenza has been kind of the broadly neutralizing vaccine, or broadly neutralizing antibody vaccine. Are we any closer to that? Because it may have then clues for what we could do around coronavirus. I mean, that's been the pursuit for a decade or more.
Sure. I mean, I think the universal flu vaccine is, I think we're getting closer. I think the strategies for that might be a little bit different than what we're probably going to be dealing with SARS-CoV-2. But, I think, thinking about, these ways of targeting a virus that, is always changing, and the ways of finding something unifying. The tricky thing with influenza that might also affect SARS-CoV-2 is it's hard to find that one antigen that will rule them all and provide protection, often because it's hard to direct the immune response against those very conserved regions of the virus. Those universal targets are sometimes elusive, and I don't know if we know how it'll play out with SARS-CoV-2 yet.
Let's go through some of the questions, and I always tell the people who I'm talking with, you have the right to say you can't answer that question or you don't know. Okay,
you'll hear that a lot.
It's not like Jeopardy that you got to answer the question. So how do you explain that children seem more vulnerable to some of the new variants?
Yeah, I've seen mixed data on certainly from the UK early on, it was thought that there was a lot more spread of the new variant, of the B117, the quote UK variant. A lot of those things have been rethought as data comes in. And so I say we're still not sure, or at least I'm not sure, given my knowledge of the differential between kids and adults. So I think we'll learn more as the weeks and months pass.
We had published a research letter very early on about the amount of ACE receptors in the nose of children versus adults, and it does seem to vary because certainly children have been much, much less prone to serious disease.
Sure.
And that's very different than flu, which is very interesting. I mean, the question has always been, do children spread COVID-19 more effectively than adults? But they certainly get less serious disease, at least pre-pubertal children. I'm always hesitant. I'm a pediatrician, so when we say children, I think we really need to talk about pre-pubertal versus post-pubertal.
Right. And I'm an internist, so I say, I don't do kids.
You know,
so
right? You know,
pediatrician.
Are there any studies being done on viral shedding from people infected with different variants, or are those studies being ongoing?
Yeah, I think they're ongoing. There have been whiffs of data that suggest that there are differences in shedding. Those studies are very hard to do because you have to control for the time-varying nature. When you sample someone in the course of their disease can affect the amount of virus you detect. And so, those studies are ongoing, and some do suggest that there's different levels of shedding with the B117 variant versus, what was before.
I don't know if you had a chance to read Paul's viewpoint in JAMA, but the question is, do frameworks for the proposed surveillance systems and repositories proposed in Paul Offit viewpoint exist in the U.S.? So what Paul and I had discussed before he wrote it was that there be a single place. Obviously, we would favor the Centers for Disease Control that could collect sera and the variants and then report out what was occurring at the national level. Does that make sense to you, Adam?
I think it makes sense. It's what's largely done with influenza virus for years. The CDC has the surveillance networks and collect viruses and sera from all over the country with academic and hospital partners. The NIH also has parallel efforts, and so I think those sorts of I think those sorts of plans are underway. And so I think it'll probably be, certainly an organizational structure from the federal government, but probably in partnership with, research institutes and universities and across the country as well.
Is there any reason to be hopeful or to think that the South African variant, the UK variant, I know people are using the numbers, and Rochelle Walensky asked me to use the numbers and not the names of countries because it can be stigmatizing. So I do apologize. It's hard for me to remember all the numbers. Is there any reason to think that all those variants won't make it to the United States at some point?
Well, they'll have.
Okay.
So B117 is I think there's at least been close to 500 sequences now
in the United States.
Yep.
Because that's one of the questions. And B117, is B117 from the UK or South Africa?
That's the UK variant.
Okay.
We've had, I think, 30 documented in Michigan, where I live.
All right.
And then P1, which is the Brazilian variant.
Brazil, right.
Was,
Thank you.
I believe, identified recently by the state health lab in Minnesota. And then I believe the South African variant, which is B1351, there were 2 cases that I know of in South Carolina. And then, as we kind of discussed earlier, the concern is we don't do as much sequencing in the U.S. relative to the UK. And so we may be just seeing the tip of the iceberg. It might be reminiscent of earlier in the pandemic when there wasn't as much testing. And that there was, circulation that we didn't know about until later. So I think that's the concern. certainly I think B117 is here for sure, the UK variant. The other ones, it's unclear whether we're containing them or not, and so we don't know how widespread.
Is the testing for the variant? Before we started, you said you were at the University of Washington, UCSF. I'm more familiar with much of Boston medicine. Does every city, every major hospital, academic medical center have the capacity to test for variants? Is it too complicated, or are there enough centers that could do it so that you could easily have 20, 30, 40, 50 centers that are testing for variants if they had the resources?
I think that model would work. There is this CDC has a Spheres. It's called the Spheres network. It's kind of a network of state labs as well as academic labs. My lab's part of it. Many other academic labs are. I think there's the problem. I think to date has been it's patchy, right? We do a ton of sequencing in Ann Arbor, Michigan, but that's great in Ann Arbor, Michigan, but it's not telling you what's going on in Chattanooga, Tennessee. And so what we need is probably more widespread. And I think these kind of federal, state, research institute partnerships would be great. And you could easily see how we could, with a fair bit of organization and logistics, get this done. And then funding, of course. And a lot of it is figuring out how to get the samples from the labs to the place where they're going to get sequenced and how that's linked to the data that you need to start making sense out of it.
Can you imagine, given the emergence of variants, that someone is going to get a Moderna vaccine and a Pfizer vaccine, or a Moderna vaccine and a J&J vaccine? At the moment, we're in the U.S. we're committed to Moderna ×2, Pfizer ×2 in a very, very specific time sequence, give or take a few days. But Pfizer's 3 weeks later, Moderna's 4 weeks later. J&J is likely to be approved in the coming months, given the press releases. It could be within a few weeks. But can you imagine that there'd be a mixing and matching that may evolve over the next 6 months or a year, or not likely?
I can see it at some point. I mean, this is kind of where we reside with influenza these days, that there's, the various egg-based vaccines, there's cell-based, there's, subunit vaccine. There's all these different ways that people make flu vaccines. And I can see us getting to that place. Whether that's going to be driven by the variants or not, I'm not sure yet, because right now they're all kind of targeting the same protein. The genetics are the same. But we may get to a point where, that maybe some of them are updated sooner than others, and it's a real complicated, both public health and regulatory problem to think through.
A couple more questions. Many more questions have come in. Would it be better for immunocompetent people to get live attenuated vaccine, as it will expose the body to the whole virus, rather than single proteins? I mean, that we've been focused on adenovirus, adenovector, and mRNA, but there have been other developments. I'm not entirely sure what both vaccines from China are, but would a live attenuated vaccine likely provide broader protection?
It certainly might, or it might potentially stimulate more immunity kind of at mucosal sites, which is, perhaps of interest for a respiratory virus like SARS-CoV-2. Obviously, there are always, kind of regulatory concerns with a live attenuated vaccine. And boy, are you sure it's attenuated? Are you sure it's stable? Those sorts of things, I think, would give people pause, but it's certainly something that people are investigating and is worthy of investigation.
Where are we when it comes to modeling possible escape mutations in the spike protein?
Sure. So
can you explain that question for our listeners?
Right. And so the idea would be that, these variants have a lot of them have mutations in the spike protein, which is what's predominantly targeted by our immune system, and it's what's in the vaccines. And so the idea is if a virus has a escape mutation, a mutation in spike that allows it to escape antibodies, right? What we were talking about earlier, that virus may escape our immune system and still cause infection despite vaccination. And so a large part of what we were talking about with sequencing is trying to see if we're seeing hints of that, and seeing if in a vaccinated population, are you starting to see more of viruses with certain mutations, because that might provide a clue that those viruses are having an advantage in escaping.
Adam, you're so knowledgeable about this. So I really want to thank you for joining me. But a birdie has told me I have to ask one final question.
All right.
Could you tell me a little bit about your birthday cake from September? Apparently, we have an image of it, and I have been told it was quite interesting. So could you say a little bit about what we're going to
Sure.
see, if we're going to, hopefully we're going to see it?
This is kind of a surprise. I guess I got clued in a little bit earlier. But yeah, we had a coronavirus a birthday cake. And so, I guess it's making the best. I have a September birthday, so before a lot of the variants emerged, making the best of a pandemic.
Who made it for you?
I would need to see the picture, because there were several cakes around.
Okay.
And so there was also a former postdoc in my laboratory who was a fantastic baker and would make a lot of cakes. And so that was always a treat for our laboratory birthdays.
Did your kids and your wife love it?
Yeah, my kids are, they're of course, indoctrinated into virology, and so they know more coronavirology than most adults at this point, for better or worse.
One last question. So Tony wrote a viewpoint for us almost 3 years ago about the speed at which vaccines were being developed. Mm-hmm. And he has this wonderful table in it: 20 years, 15 years, 10 years, 8 years. I think he finally gets it down to like 3 years. And this was 3 years ago. Yeah. You're 10 or 15 years into your career. Did you ever imagine that you would see a virus sequenced in January and there'd be testing of a vaccine in June or July?
It's bonkers. Yeah, it's amazing. And I think it's one of the things you can lose sight of in the moment we're in. It's, because there's so much concerning and bad news, and things are hard. But then you're just like, wow. I mean, it's remarkable. From sequence to 2 EUA vaccines that are highly effective within a year. That's just amazing. And the other thing to think about is, people are concerned about the variants, and, are the vaccines going to work as well? There was data last week, from Novavax, and then some data from the Janssen vaccine that, 50 to 70% against the variant, perhaps. Boy, if we got 50 to 70% vaccine effectiveness for influenza every year, we'd be ecstatic.
Right. It's so interesting you say, because I think the struggle has been, the bar. I think when people saw 95%, firstly, I think Pfizer was first. I can't remember.
Yeah.
And then 95% from the other one. It was so nice that the same vector platform, mRNA, came up with the same. I don't think people they were disbelieving. And so now the problem is when you have new vaccines and they say 70% or 60%, people are going to go, oh, my Lord, it's only 60 or 70%.
Right.
There are very few vaccines against viruses that approach 95%. It was an extraordinary achievement, and the bar has been set so high.
Right, or to the Super Bowl analogy, the goalposts have been moved, or our expectations, because it was just amazing how well those first two reported vaccines worked. And so, it's, that's when I start getting concerned and kind of getting down on this, that's kind of what I come back to. And we'll see how it plays out.
This is Howard Bachner, editor-in-chief of JAMA. I've been talking with Adam Lauring. Adam is an associate professor of internal medicine at University of Michigan. He has a position in the Division of Infectious Diseases, Microbiology and Immunology, and Ecology and Evolutionary Biology. And almost a month ago, anticipating the struggles that we may have with variants, Adam, along with Emma Hodcroft, wrote a viewpoint for us entitled Genetic Variants of SARS-CoV-2, What Do They Mean? Adam, thanks so much for joining me today. And I didn't get a chance to see the picture of your cake, so now I have to go watch the video so I can see a picture of these cakes.
Indeed. Well, thanks for having me. This is
Thanks,
great.
Adam. Take care.